Hi student, I am Dr. D.S. Thavre. Now I share the screen of today's lecture in front of you. Welcome to the fourth lecture of course of fifth semester, that is PAP 3.4. Title of this course: Disease of Field Crops and Horticultural Crops and their Management. Today we discuss about downy mildew diseases in field crops. So this is the only two diseases seen in pearl millet and maize, respectively by Sclerospora genera, means downy mildew of bajra caused by Sclerospora graminicola, downy mildew of maize caused by Sclerospora philippinensis or Sclerospora madis. So see the first disease seen in pearl millet or bajra caused by Sclerospora pathogens causing downy mildew disease. So downy mildew infection is mainly systemic in nature. Means infection generally start from the initial growth stage of the seedlings. So this infection mainly spread from seedlings to the well-developed plants, especially in airheads. So these downy mildew pathogens develop the symptoms on particularly leaf of the bajra and airhead also. So initially the downy mildew pathogen develop the symptoms at seedling stage of the bajra at 3 to 4 leaf stage. So see this infected individual leaf of the bajra. So this is the light green patches as well as light yellow patches seen on upper side of this infected leaf of the bajra. So this is the initial infection majorly seen on upper side of this bajra leaf. Also seen the lower side of the infected leaf of the bajra. So this is the whitish downy growth of this fungus can be seen on the lower side of the leaf. Means you know about the which type of the symptoms produced on which side of the leaf. Means generally chlorotic patches or chlorotic streaks mainly seen on the upper side of the leaf and corresponding lower side of the leaf having whitish downy growth may be seen. So under favorable conditions, this yellow discoloration turns to the brown in colors, means long streaks generally seen along the leaf vein. So this is the symptoms generally seen on the upper side of the leaf and this is the lower side of the leaf. Then you see these infected seedlings. So this infected plant produces the maximum tillers from the single seeds, and these tillers cannot develop the airheads. means the infected tillers drives means completely plants will be dries because they reduce the internode size of the stems. So infected plants is always small height means stunted the growth of this infected bajra plant. So this symptoms is also seen in the bajra plants infect due to the downy mildew pathogens. Then most important symptoms generally seen on the airhead portions because this disease is systemic in nature. So that's why the majorly symptom seen on the airhead portions means the infected plants develop the airhead or sometimes is felt to the develop the airhead. Suppose this infected plant develop the airheads, so partially or completely transformed these ovaries into green leafy structures. So see these first pictures. So these infected airheads is partially converted into green leafy structure. So you see these green leafy structures means after infection of the pathogens to these developing ovaries, so these floral parts is totally converted means the blooms, the pali, stamens, the stils. So this is the floral parts. So these floral parts is completely converted into green leafy structures. So these green leafy structures become brown in colors and dry bearing masses of the oospores means finally develop the spores. So this is the infection generally seen on the airhead portions. So see these diagrams. So in these infected airheads, the complete transformation of the reproductive organs or floral parts convert into green leafy structures. So this green leafy structure become dries and having number of the spores produced, that is oospores are produced. So this picture in which maximum number of the infected bajra plants are shown. So these bajra plants in which the complete transformation of the airheads into green leafy structures are seen. Okay, so this is the symptoms produced by sclerospora pathogen in pearl millet or bajra. Okay, means this fungus develop the symptoms on leaf portion as well as airhead portions. Suppose this infection seen on the leaf, so majorly develop the symptoms on both side of the leaf and airhead portion. The airheads sometimes partially convert or sometimes completely convert into green leafy structures. Okay. Then we go to the next point, that is a causal organism, that is Sclerospora graminicola. So this fungus is responsible to produce the downy mildew disease in pearl millet or bajra. Okay, then etiology. Etiology means morphological and taxonomical characteristics of a particular pathogens. So this fungus develop the mycelia. So this mycelia is always sinuosity or non-septate and intercellular in natures. So this Sclerospora fungus reproduced by two methods. One is the asexual methods and another is the sexual methods. Now you see this disease cycle of this Sclerospora pathogens means this is the infected bajra plants in which develop the downy mildew infection on particularly airhead portion as well as the infected leaf. Means after harvest of this bajra plant from a particular field, so number of the infected leaf or maybe airhead fallen in soil. So at the time of the flowing operation, these infected plants, especially leaf and airhead, debris in soil. Means this fungus goes to the dormant conditions and develop the mycelia. But after some days or under favorable conditions, these dormant mycelia develop the sexual spores that is known as the oospores. Means before the formation of the oospores, these dormant mycelia develop the oogonium. Oogonium means female gamete and antheridium means male gamete. So fusion of male and female gamete under meiosis. So finally develop the oospore. So oospores is a sexual spores. These oospores generally spherical in body, having two-walled. So these oospores always present in soil. Means in next year, suppose the farmer can sow the seed in particularly fields. So these seeds become germinate and produce the seedlings. But at this time, the number of the oospores present in soil and these spores directly infect to the germinating seedlings. Means these oospores firstly infect to the seeds as well as roots of the seedlings. So this fungus become persist along the growth stage of these seedlings. So this infection is initially developed on the producing the leaves. Means the chlorotic patches and white downy growth may be seen on this leaf. So this fungus become systemic in nature means develop the symptoms on these producing the airheads. So these airheads is completely or partially transformed into green leafy structure. Means already symptoms is produced in leaf or airhead portion. Means the sporangia and sporangiophore is developed in the infected leaf or infected airheads. Means this mycelia develop and produce the sporangiophore. So at the tip of the sporangiophore, they bear the number of the sporangia. So this is the sporangia. Means this sporangia is produced at the tip of the sporangiophore. Means already disease is produced. So this sporangia is released or liberated from this infected airhead or infected leaf and fallen on the healthy one plants through wind or air currents. So these healthy one plants become infected. So in this way, spread the disease from infected plant to the healthy plants. Means in short, primary infection is takes place through oospores is survival in soil up to 8 to 10 years. So this is the one possibility to initiate the primary infection. Then another possibility, the suppose the farmers can use the seeds from infected plants, and this infected seeds is used for sowing purpose in next year, so 100% disease is produced. Means infected seed and oospore present in soil, these two possibility to initiate the primary infection in healthy one plants. Then actually secondary infection takes place through this sporangia and these zoospores. So these zoospores and sporangia is disseminated through wind or maybe through water and deposit on the healthy one plant. So these healthy one plants become infected. Means in this way spread the disease from healthy one infected plant to the healthy plants. Okay, so this is the disease cycle of the sclerospora pathogens. Okay, so this is the oospores already developed through the contact of the oogonium and antheridium. Oogonium is a female gamete and antheridium is a male gamete. So these oogonium and antheridium fuse to each other. So finally develop the sexual oospores are formed. Then the this is the infected leaf, means already spores are developed. So these spores generally penetrate to the healthy one leaf or healthy one airheads, means develop the mycelia in intercellular mycelia. Intercellular mycelia means mycelium generally grows in between the two cells. Means this is the one cell, this is the another cell. So this is the just mycelial network is seen in between the two cells. So this mycelia emerge out from the infected stroma and produces the sporangiophore. So this sporangiophore is unbranched up to 3/4 portion of this sporangiophore, means this sporangiophore is unbranched in base portion. So at the tip of the sporangiophore, the sterigmata is produced and this sporangia generally produced at the tip of this sterigmata. So this is the papillate form of this sporangia is produced at the tip of this sporangiophore. So this sporangia again germinate under the favorable conditions and to produce zoospore. So the zoospore is a biflagellate type of the spores. Biflagellate means two flagella is present and always kidney shaped. So these two spores directly infect to the healthy one leaf. So this healthy one leaf become infected. Okay. Then now go to the next point, that is a favorable conditions. Means first favorable condition for germination of the spores, that is oospores as well as the sporangia, that is a high humidity, means maintain the relative humidity is near about 90%, then presence of water on leaf, then temperature, that is a 15 to 25 degrees Celsius. This is the climatic factor is responsible to produce the disease in pearl millet. Then one soil factor is also considered for development of the downy mildew disease in pearl millet, that is a ill drained soil. So this is the one factor is mostly infect or favorable to produce the downy mildew infection in bajra crops or pearl millet crop. Okay, then goes to the last point, in downy mildew disease of bajra, that is a control measures. So you know the primary infection takes place through use of the infected seeds. So avoid this problem means you always use the disease free seeds or select the seeds from healthy one crops in particular field. This is the one control measure against the downy mildew pathogens. Then collect the diseased plants, especially before the formation of the ooze spores, because ooze spores acts as a primary infection to the new one healthy one plants. So this locate the infected plants and properly burn. Then summer deep flowing practice should be done because these ooze spores mainly survival in the absence of the main host up to 8 to 10 years. So break down the chain of these ooze spores or eradicate the ooze spores by adapting the flowing operations. Then rogue out the infected plants. Rogue out means locate the infected plants and uproot from this place. Then prolong the crop rotation with non-host crops. Means you do not cultivate the bajra crop year after year in the same fields. Means to avoid the infection of the ooze spore by growing the non-host bajra crops. Then treat the seeds with metalaxyl at the rate of 3 to 4 gram per kg or Thiram or Captan is also effective and use at the rate of 4 gram per kg of seed. Mean to protect the seed from the fungal infection by applying the metalaxyl or maybe Thiram or Captan fungicides. Suppose the infection generally seen on the leaf portion of the bajra or airhead portion of the bajra. So immediately spraying this crop with mancozeb at the rate of 0.25% or metalaxyl at the rate of 0.2%. So these sprays should be done starting from the 30 days after the sowing in the field because this infection majorly seen in seedling stage also. So avoid this infection at the seedling stage. So first spray should be done 30 days after the sowing in particularly bajra fields. So this is the some cultural and chemical management methods or just a chemical uh, measures should be applied to avoid the downy mildew disease in bajra. So instead of these management points, you can consider regarding the resistant variety, means grow resistant variety or moderate resistant variety like Mallika Arjun, then Jain Bajra, then PHB 14, then PHB 10, ICMH, then ICPT 8203 or WCC 75 means some cultivars as well as some genotypes is a resist against the downy mildew infection in bajra crops. Okay, so keep in your mind the information of downy mildew disease in bajra. Okay, then now we go to the next disease seen in maize caused by Ulcosclerospora and produces the downy mildew disease. So this is the infected leaf of the maize plant. So initially the chlorotic streaks is seen on the particularly leaf portions, means on the upper side of the maize leaf, just the chlorotic streaks is seen. Okay, so this is the initial infection seen on the upper side of the leaf. But you seen also the lower side of this maize leaf. So this is the whitish downy profuse growth may be seen on the corresponding to the lower side of the maize plants. Means this is the upper side of the maize leaf and this is the lower side of the maize leaf. Means after favorable conditions, these chlorotic patches or chlorotic areas may be increased over this green color of this leaf. So finally, this infected tissue, generally brown in color, or sometimes this infected tissue may be dries and such infected leaf is seen on these infected maize plants. Okay, so this is the infection seen on the upper side of the leaf and lower side of the leaf. Okay, under favorable conditions, these downy mildew pathogens also be infect to the male flowers as well as tassels. Means the infection generally occurs on bract up green unopened male flowers in a tassels. So these tassels is converted into green twisty leaf. So this male flower and tassels is completely converted into twisted green leafy structures. So these symptoms majorly seen in the maize plants. Okay, means under favorable conditions, this infection is start from leaf to the airheads. Means before the actual reach this infection to these airheads, the infection generally seen in the leaf portion. Means in high inoculum load of these spores develop inside the leaf. So leaf may be shredding, means shred the infected leaf. So this type of the symptoms also be seen in the maize plants. Okay, means the proliferation of auxiliary buds on the stalk of the tassels as well as copes. These symptoms is very common seen in maize plant due to the downy mildew pathogen. So this type of the symptoms is also known as the crazy top of the symptoms. Okay, means the plants exhibit the stunted and bushy appearance. So these bushy appearance symptoms generally seen in maize plant due to the shortening of the internodes. Okay, so this is the symptoms seen on male flowers as well as tassels or copse portions. Okay, then causal organism Sclerospora philippinens or Sclerospora madis. So this fungus develop the mycelium and reproduce by sexually as well as asexually. Suppose these Sclerospora genera reproduce by sexually methods. Means finally this type of the oospores are developed. So oospore is a sexual spores developed after the fusion of oogonium and antheridiums. Then this fungus also be developed or reproduced by asexually method, means the mycelia germinate under the favorable conditions and to produce the structure. This structure is known as the sporangiophore. So this sporangiophore is unbranched up to the 3/4 portion of the sporangiophore. Means branching structure is always seen at the top portion of the sporangiophore. So at the tip of the sporangiophore, this type of the sporangia is generally developed or produced. So such sporangia is liberated from the stoma and directly infect to the healthy one plants. So these healthy one plants become infected. Means sporangia develop through asexual methods. But under favorable conditions or with the help of the moisture, these sporangia again germinate and to produce the zoospores. Okay, so this is the sporangiospore, unbranched up to the 3/4 portion of the sporangiophores. So sporangia is developed at the tip of the sporangiophore. So this is the papillate form of the sporangia is produced at the tip of the sporangiophore. So this sporangia is developed asexually method. Means zoospores and sporangia acts as a secondary infection through moisture or through winds and infect to the healthy one plants. So these healthy one plants also infect. So in this way, spread the disease from one plant to the another plant through sporangia and zoospores. So this is the oospores. So this oospore is a sexual spores. And this oospores is always present in soil for more than 8 years without a host. Okay, so this is the perpetuation and disease cycle of the sclerospora. Then favorable conditions, the high relative humidity that is above 90%, then temperature range that is 20 to 25 degrees Celsius, then younger plant is highly susceptible to this disease as compared to the older plants or mature plant. So this is the some factors to be considered for development of the disease in palm millets. Okay, so now next point, how to control the downy mildew disease in maize. First one, clean cultivation, because the primary infection takes place through oospores present in soil. Means after harvest of the crop, the infected leaf or infected airhead of the maize may be fallen on the soil. So at the time of the flowing operation, these infected plants debris in soil. So it is necessary to clean the cultivation to avoid the primary inoculum developed in particularly field. Then crop rotation with pulse crop. Crop rotation means they do not grow this maize crop year after in same fields. Means this crop rotation with pulse crops. Then removal and destruction of collateral host. means some uh, collateral host. For example, sorghum bicolor, sorghum halpenins. This collateral host acts as a survival of the spores in the absence of the maize crop. Then this fungus also be survival on weed crop, that is a Digitaria weed species, acts as a collateral host for Sclerospora pathogen. So eradicate the inoculum of the Sclerospora on collateral host. So this practice is necessary to avoid the inoculum on the collateral host. Then seed treatment with Ridomil or Metalaxyl at the rate of 4 gram per kg of seed, because initial infection is also takes place through use of the infected seeds for sowing purpose. Then avoiding areas prone to flooding and providing for adequate drainage of surface water. So this is the best method for controlling disease effectively, because the Sclerospora fungus majorly produces the symptom in ill-drained soil. So avoid such ill-drained soil for cultivation purpose to the maize. Then spray the crop with Titan M45 at the rate of 0.3% or Ridomil at the rate of 0.2% or Metalaxyl at the rate of 1 kg combined with or combination with mancozeb, means metalaxyl plus mancozeb. This is the combination product. This combination product is known as the Ridomil. So Ridomil generally most effective to downy mildew fungus in maize. So this Ridomil can apply at the rate of 1 kg per hectare at an interval of 10 days. So this spraying should be start from 20 days after sowing. And last management point, that is a grow resistant variety or tolerant varieties. For example, DHM1, DHM103, DMR5 and Ganga second. So this is the some management or some control measures should be done every farmers to avoid or eradicate the downy mildew disease in palm, in maize crops. Okay, so today's we discuss two diseases, that is a downy mildew of bajra and downy mildew of maize. Okay, so any doubt about today's lecture, so you contact personally to me. Okay, any doubt? Okay, now I end up these lectures. Okay, thank you.